So I have a couple of handouts for you. These are some practice problems to go over really quickly. Hopefully they won't take too long. These are related to your homework for the week, or for next week. Your homework will be due next Monday on the day of the exam for chapters 5 and 6. Next Monday for the exam, it's going to cover chapter 17, which is the one that we did sort of online when we canceled class for a reason that I don't remember at the moment. So the next exam will be chapter 17, chapter 5, chapter 6, and then it's scheduled to also be 7. Depending on how much we get through for 6 today, 7, well, I'll let you know about 7. So we'll say tentatively chapter 7. The lecture for 7 is pretty short, so we might get through it, we might not. I'd like to keep it on this exam if we can, because otherwise the next one's going to be like the first one where it's just really big. So hopefully 5, or for sure 5 and 6, hopefully 7 also. And then homework will be due on test day. And then also in Brightspace, I posted a extra credit assignment. I posted it several weeks ago. It goes with chapter 5 with mapping. So it's just a short article to read and some questions to answer. So if you want to do that, that will also be due on test day as well. So, it's been a while since we've had a lecture. Do you all have any questions before we get started? I feel like we're just making our rounds around the biology classrooms. I don't know where you all are supposed to be sitting. I know where you're supposed to be sitting in the other rooms, but this one's different. Actually, last spring we had genetics in here. I actually really like this classroom. I like the way it's laid out. Yeah. It was terrible for genetics because we don't have anything in here for genetics, but we had to cart it in from all the other places. So anyway, all right, well, we'll go ahead and get started. So chapter 6, we're in the middle of talking about our chromosome structure alterations. So there are 3 different structural alterations. There's chromosome rearrangements. We started talking about those already. We'll wrap that up. And then there's also aneuploidy, which is where we alter full chromosomes. So we have an extra or a missing chromosome. And we also have polyploidy, which is where we have duplications of the entire genome. So we'll cover those 2 in just a minute when we finish up chromosome rearrangements. We have already talked about duplication and deletion. So sometimes those happen in tandem. Kind of depends on the actual crossover event. But if you're looking at crossing over in meiosis, lining up, usually what happens is that one chromosome has a duplication, the other one has a deletion because recombination happens and moves a chunk of the chromosome. I believe we already talked about inversion, is that correct? Or we flip pieces of whole sections of chromosomes. I believe we talked about that. I think so. Okay, hopefully. All right, so we need to go through translocation. And so we're going to start there. But does everyone have inversion in their notes?
Yeah.
Okay.
I have inversions in meiosis can result in loops to pair affected regions.
Okay, perfect. So we're going to do translocation, but real quick, I have a set of practice problems that we can go over together. Jacob, I think you had asked about doing some chromosome practice problems. We hadn't quite got to them yet when I handed out the homework. So in your homework for this week, you'll have a chromosome, 1 or 2 chromosomes that is an original sequence. So what this means is that you've got a chromosome with a centromere in the middle, and then you've got a sequence of genes or loci along your chromosome. Usually a star or sometimes a dot in the center just indicates the centromere. And then we've got our other genes on the other side of the chromosome. So in your homework, you're going to be presented with sort of a list of genes in order along the chromosome, and you'll have to figure out what has actually happened to that chromosome. So sometimes there are multiple terms that are going to apply. So you're kind of looking for, so you're looking for basically everything that could have happened to the chromosome. So, did I hand out to somebody the one with my notes on it? I think I did. Oh, is this one? Yep, that one. I'm gonna hand this back to you. Let's switch out. All right, great. Okay, so you guys have 2 copies, a duplication deletion practice and an inversion translocation. We'll get to that one in a little bit. But for our duplications, there are several different types. We've got tandem, displaced, reverse, and segmental. And then we've also got just regular deletion. So deletion is just going to be when any chunk is removed. There's not really any specific term for that. But just as a quick review, tandem means that you get 2 of the same chunks adjacent to one another. So you get a duplication and then the same chunk right next to it in position. Displaced means that you have a duplication that is somewhere else, so not adjacent to it. Reversal means that when it was inserted into a new location, the sequence is now backwards. And then we've got segmental. Now segmental is basically, it just means it's a big chunk. So you won't really see that represented in these just because you have no way of really knowing how many base pairs. I believe it was defined as more than 1,000 base pairs. So you won't be able to sort of see that in this list. But if we look at the first one, can anybody identify whether this was a duplication or a deletion? Duplication, very good. So which part was duplicated?
C.
Very good, CDE. So this is our original segment. This would be our duplication, or the other way around. We don't really know which one is the duplicated one. So it's a duplication. What kind of duplication? Is it tandem, displaced, reversal, or segmental? Tandem, very good. So this is going to be your tandem duplication. All right. So you're really just looking at what happens to those letters. Just keeping in mind that those letters are referring to a region or a particular gene on your chromosome. All right. Second one, duplication or deletion?
Duplication.
Duplication again, very good. What kind of duplication?
Reversal.
Reversal, anything else?
Tandem.
Very good. So this one, there's a whole lot going on. So we've got the IJK, KJI. So this is a duplication. It's tandem. And it's a reversal. I'm talking kind of loud so they can hear me over there. I don't know if there's a microphone over here or not. All right, so any questions on these ones so far? All right, how about that bottom one? What happened there, duplication or deletion? Deletion, very good. So in this one, we deleted the centromere from the original, F and G. So right about here, we have a deletion. We didn't talk about any specific types of deletion, so deletion would be appropriate here. When we looked at inversions, we talked about a paracentric inversion. This doesn't apply to this slide, and a pericentric inversion. These are terms that reflect that the centromere is involved. But I'm not aware that you talk about deletions in reference to the centromere. So we've got a deletion here. We've deleted that centromere. So what's going to happen during meiosis if there's no centromere there?
It won't like get transported with the rest of the chromosomes.
Very good. So yes, without that centromere there, it won't get, it might line up, but it's not going to get pulled apart during mitosis, meiosis, and eventually that chromosome is going to be lost. And so you're going to end up, when you lose a chromosome, this falls under the category of aneuploidy. So the result of losing this centromere here is that you're going to lose a chromosome during meiosis. All right, so if you have not started chapter 6 homework yet, that's perfectly fine. Hopefully this can get you started. Once we get through inversion and translocation, I've got another set of practice for you. So we'll talk about those in just a minute. Besides my typos on the slide, are there any questions? All right. So we are going to go ahead and move on with translocation. So nothing earth-shattering about the name here. Translocation is just means that we're going to take a chunk and move it to a different or opposite location. So the difference here between duplication or deletion is that we're transferring between non-homologous chromosomes. So you may have a displaced duplication. When you have a displaced duplication, it's happening along homologous chromosomes. So even if your duplication happens with a deletion, so you have two chromosomes, one ends up with a duplication, one ends up with a deletion, it's happening across homologous chromosomes. When we introduce translocations, what's happening is you have two chromosomes that are a different size, different shape, different genes, and those chromosomes are lining up and recombining, even though they don't fit our definition of non-homologous. So we're getting two different chromosomes, and maybe they're lining up because they have regions of similarity, that maybe the sequences in that particular region are close enough that the chromosomes sort of get confused and line up even when they shouldn't. And so what happens is you're going to get a chunk of one chromosome moved over to the other in the non-homologous. So we might end up with something like so depending on the exchange, it might be reciprocal. meaning that both chromosomes exchange a piece, or it might be non-reciprocal, meaning that one chunk moves over, but nothing takes its place. So we'll see in just a minute. There are 3 different kinds of translocations, depending on how the exchange actually is working. All right, so we've got 3 different types. We've got non-reciprocal. Basically, this means that there's no exchange of information. So you get a chunk that's moved from one segment to another. This one could happen due to recombination. So if your chromosomes are lined up incorrectly with the wrong type of chromosome pair, then you're not going to get an equal exchange. But more often for non-reciprocal translocation, what happens is that there's a chromosomal break. And the cell attempts a repair. So basically what happens here is something causes the chromosome to break. Maybe this is damage to the cell, maybe this is UV damage, something like that. And as the cell's getting ready for mitosis or meiosis, it attempts to repair. So remember, one of our checkpoints during meiosis is the S-phase checkpoint to make sure that, or right before S-phase, sorry, to make sure that our chromosomes are ready to go, ready for replication. And then again, after S-phase, we have another checkpoint to make sure that everything has been replicated. So if the cell finds that there's a chromosome break, sometimes it will reattach a piece of a chromosome in a place that it's not supposed to be. So the cell doesn't necessarily know where that break happened, and so sometimes a repair happens and it gets put back in the wrong place. So if we've got the non-reciprocal, it's not so much as an exchange as a movement of a section. More common is the reciprocal translocation. So this is where 2 chromosomes come together. What did I have? I had A, B, D, E, F, G, H. So a reciprocal might be something like A, B, D, H. So here's your exchange, F, G, E. So you've got this exchange, this opposite sort of complementary exchange between these non-homologous chromosomes. So when we were talking about duplication and deletion, we talked about some of the problems that come up from this. One of them is regulation of genes. So if a gene is supposed to be expressed along with another set of genes or under the control of a particular gene expression mechanism, that gene may not be expressed correctly now that it's over on a different chromosome. Also, depending on where the break happens, if it interrupts a gene, you might now have a dysfunctional gene. You might have a loss of function if that gene is sort of broken in the middle. Because remember, all of these duplications, deletions, all these crossing over are happening at almost at random within the cell. And so it's not necessarily checking to make sure that it's is or is not disrupting a gene. If it is lined up correctly, it shouldn't disrupt anything. But if it happens to be lined up incorrectly, it might. So you got regulation happening. And then, of course, if you just lose the function. All right, our last kind of translocation is called a Robertsonian translocation. So this is kind of a specific type of reciprocal translocation. In a Robertsonian translocation, what happens is you get basically the two long arms coming together from two opposite chromosomes. So let's say we have, nope, that's not a good chromosome. Let's do something like that. What might happen here is that you get your two long arms combining and your two short arms combining. And you end up with a little tiny piece of a chromosome. And then the majority of the other chromosome still intact, but connected to another. And so you end up with this little tiny fragment that still potentially has a centromere, but those little tiny fragments are really easy for the cell to lose. And so with a Robertsonian translocation, sometimes what happens is when you get this reciprocal translocation, you lose part of your chromosome.
Do they get lost just because they're so small?
Yeah. I think I'm missing a slide. A minute. Oh, never mind. I know where it is. Okay. It's just not right here.
Do they have to be significantly smaller than the long arms, or can they be like pretty close but still considered smaller?
I don't know what the cutoff is for like smaller. My guess is that there are different like lengths. Like this could happen. I think what the point is here is that you basically get most of two chromosomes and hardly any of the other two. But I don't know like where the middle ground is, like when it's considered small. I think it's basically considered small if it's potential to get lost.
Okay.
But I don't have a good answer for that. Any other questions? So I already mentioned this. This is just a diagram, though, that shows what a translocation might look like, much better than my diagram. So what's happening here is it's showing that during meiosis, our sister chromatid pairs line up, but they line up incorrectly. And this is probably because there's some region here or maybe here that is very similar. The reason that they might be similar is because through the process of evolution, duplications, deletions, translocations, all of these things have probably happened at some point. And then those sequences begin to diversify. The sequences take on different functions, the genes evolve. And so there are probably actually sequence similarities there. There has to be enough similarity that they line up. And then any region that is different just sort of gets pulled along for the ride. In this image here, this is showing kind of what happens when the chromosomes line up and exchange information. So you can kind of see once they get pulled apart where they're going to end up. So we get lining up during meiosis. Then when they separate, because they're lined up incorrectly, they take the wrong pieces with them. All right, so we talked about the 4 big chromosome alterations. There are 2 other types. There's a few other types of chromosome variations, but the other 2 that are textbook, oops, I'll come back to this in just a second. The other 2 that our textbook talk about are called fragile sites and copy number variations. So these are not anything to do with crossing over or homologous chromosome pairing or non-homologous chromosome pairing. These are basically structural, well, this one is a structural problem. A fragile site is a structural issue in the makeup of the chromosome. So a fragile site is a region that is prone to breakage. So under certain conditions, for example, you might have a piece of chromosome break off. These tend to happen at specific regions, and it just depends on the molecular structure of the DNA. Maybe it's at a point where we have lots of, maybe there's some damage or we've got some modifications to the DNA structure that basically weaken the backbone. So there are certain sites that are prone in the human chromosome. There's one on the X chromosome. This one is called Fragile X syndrome. So it happens, it shows up in males because they have only the one X chromosome. So if something happens to that X chromosome, they don't have the other one as a backup. And so any information that's at that fragile site, if it breaks off, that genetic information is lost. We'll see a couple other chromosome disorders in a few minutes. Usually these chromosome disorders are going to have lots and lots of problems associated with them. So we have our mutation disorders that we talked mostly about in chapters 2 and 3. Our mutation diseases like sickle cell anemia, things like that. They usually have a specific set of phenotypes. They may or may not have a major impact on the individual. It just depends on what the gene is for. But usually the chromosome structural alterations are going to have a big impact because they are affecting a large area of the genome. So this one, the big thing that this one causes is intellectual disability. There's a few other phenotypes that are associated. You can kind of see on this image here, it's a cartoon image, but there's kind of an elongated forehead. There's usually multiple phenotypic and very often intellectual disabilities as well. When you start bringing in the sex chromosome, sometimes you get sexual characteristics affected as well. All right, so our last structural variation is called copy number variation. So this is where you have a small region, usually just a few base pairs. So this is going to be a few base pairs. And this is regions where you have those few base pairs duplicated, and you may have hundreds or thousands of base pair duplications. So this is usually like 1 to 5 base pairs. It might be a little bit more, but usually it's a very small number of base pairs. And you can have up to, well, maybe up to about 1,000 copy number variations. Copy number variations usually occur in non-coding regions. So they're usually vary greatly among individuals. So, for example, a lot of our DNA fingerprinting is actually targeted for regions that are known to have copy number variations. For fingerprinting, they like to use something called SNPs, which is single nucleotide polymorphisms. So these are regions where you get a great number of variations, usually repeats that are going to be varying between individuals. Because they're in non-coding regions, they don't usually have phenotypic effects. We're going to talk about non-coding regions coming up in, I think, chapter 8 or chapter 9, when we start talking more about how we define a gene and what signifies a gene in a genome, and why we might have these non-coding regions. All right, so I'm going to go back to problem practice. So this is on the bottom of the sheet that I gave you. So here we're looking at inversions and translocations. So for our inversions, we have paracentric inversion. This occurs on one side of the centromere. And we also have pericentric inversion. And this one is going to include the centromere. And then for our translocations, we've got non-reciprocal, reciprocal, and Robertsonian. So go ahead and take a minute and see if you can figure out a couple of these. So up at the top, I've got 2 chromosomes here since we've got to include translocations. So there are potentially 2 chromosomes here. And the first one is the same as the other one that we looked at, and the bottom is a different one. All right, so the very first one here, the red letters, what does that one look like? We can narrow it down. Is that an inversion or a translocation? Inversion, very good. Which part is inverted? Yep, one side, so over here. So the IHGF, that's compared to FGHI. So is that a paracentric or pericentric? Paracentric, very good. So this is going to be the paracentric. Because the inversion does not include the centromere, so it's a paracentric inversion. All right, how about the green one, the one on the right? What is that one? So this one only includes 1 chromosome.
So it's pericentric, right, is the centromere?
Yep, very good. This one is pericentric. So we've got this chunk here has moved. So we've included the centromere there. So this is going to be a pericentric inversion. All right. So the ones on the bottom, this is including 2 chromosomes. So this is probably going to be a translocation. So this is across 2 non-homologous chromosomes. All right, so we're looking for reciprocal, non-reciprocal, or Robertsonian. For that first one, what does it look like to you?
The non-reciprocal.
Non-reciprocal, very good. So non-reciprocal, because we have the IJK moving down to this chromosome. So we get the translocation from the top chromosome to the bottom chromosome. So this is going to be your non-reciprocal. Very good. How about the orange one, the one on the right? Very good. This one is a reciprocal. So this one, these 2 regions here have moved between. So this would be your reciprocal translocation. And of course, the difference here is that we get both pieces moving, whereas on the left-hand side, we have only one piece moving. So that really just leaves one option for the bottom. What do you think the bottom one is? Robertsonian, very good. So this one I was trying to like indicate small. So I just took the smaller of the two ends. If you were to see this on an exam, I would try to make it very obvious that we were doing the small ends, but this is going to be your Robertsonian. Translocation. All right.
So,
Yes.
wait, so where did the FG go?
Oh, well, that's a great question. I think the FGH was lost to a typo. Is where I think that one went to. It should be right here. It should be FGH VWXYZ. I made these up like an hour ago.
Yeah.
So I was surprised there wasn't another. Yeah, so that would exaggerate the long end. But yeah, it should be there. Sorry. That was just a mistake. But what would it be?
A deletion.
A deletion. That was not intended to be a deletion, but I mean, if you caught it, you caught the deletion. But it was not intended to be that. But I suppose it could also happen.
Can you have all of that, like a Robertsonian translocation deletion? Like,
Yeah, so
theoretically have,
I mean, theoretically, you could have any combination of these.
It would just be a really bad day for your chromosomes.
Yeah, in practice, when you start having these major chromosome issues, the fetuses are usually not viable.
Yeah.
So,
And so you wouldn't have any of these happen outside of, well, no, because earlier we talked about a repair, so they could happen during mitosis. So that would just mean that the cell is not viable either, right?
probably, yeah. So if it happens during mitosis, like let's say you have a skin cell, probably what's gonna happen is like those two skin cells are gonna die, you're otherwise gonna be fine.
Okay.
Like you probably wouldn't even notice that those two skin cells have died.
Yeah.
But if this happens, during sperm or egg production, then you're gonna end up with major chromosome issues. So we'll talk about it in just a minute. We'll probably take a break. Even though, because it's a good break point. But we'll talk about some chromosome disorders in a few minutes. The major chromosome disorders are very, very rare because plants do okay with missing genetic information, but animals do not. And so if you have major regions deleted, whole chromosomes missing, whole extra chromosomes, all those, you end up with very widespread phenotypic effects. And usually they're not survivable. There's only a handful of them that are actually survivable. And usually the ones that are survivable have major effects. So if you think about Down syndrome, we're going to talk about that in a minute. That affects a whole bunch of different systems.
And that's an extra one. So without.
Yeah, that's an extra one. Extra is more survivable than missing.
It still causes a bunch of issues.
It still causes a bunch of issues. A lot of them are related to dosage. We talked about gene dosage earlier in this lecture. We have more copies of the gene than you evolved to have. So yeah, when we talk about these things, a lot of them are kind of theoretical, or there's one good example of them. Mostly because they're not survivable.
Is that why that one is named Robertsonian? What is that? Is that from someone that had it?
I believe it is named after the doctor that discovered it.
Okay.
So, spoiler alert, we're going to talk about this in a minute. There's two ways you can get Down syndrome. One is an extra copy of chromosome 21. The other is a Robertsonian translocation, where you end up with an extra copy attached to another chromosome. So, yeah. I'm gonna, if you guys wanna take a quick break, I know Nia, you just got back, but if you guys wanna take a quick break, I'm gonna check in on Teams and see how they're doing, if they have any questions, and then we'll come back in a couple minutes. All right. Lindsay and DJ, are you guys doing okay? Do you have any questions? All right, we're gonna take a break over here for just maybe 5 minutes or so, and then we'll get started again. Can you all hear me okay?